Hello everyone, this video is about clot retraction time or CRT. What is clot retraction? Clot retraction is the consolidation or the tightening of the fibrin clot. In primary hemostasis, the platelet plug is formed, while in secondary hemostasis, blood coagulation is complete with the formation of the fibrin clot. And this fibrin clot is the one that would undergo clot retraction. So in vivo, clot retraction is the shrinking of the blood clot over a number of days. So how does clot retraction contribute in the repair of the damage of the blood vessels? So in damaged blood vessels, clot retraction would pull these damaged blood vessels together so that they can repair the blood vessel wall. So clot retraction pulls the damaged edges of blood vessels together so that the repair can be faster, tighter, and stronger. In vitro, when blood clotting is complete, the clot normally undergoes retraction wherein the serum is expressed and the clot is denser. Clot retraction test helps to determine the time that is needed for the blood, which is inside the test tube, to form a clot, and for this clot to have its edges draw in from the side of the test tube. Clot retraction tests may either be qualitative or quantitative. Qualitative tests only detects the presence or the absence of clot retraction, while quantitative tests can give an estimate degree or the amount of the retraction. An example test under qualitative is the Hirschsprung method, while under quantitative is the Stefanini method, also sometimes referred to as the Stefanini-Dameshek method, and the second method for quantitative is McFarlane method. The Hirschsprung method is also known as the castor oil method because we place castor oil in a test tube. After that, we add a drop of blood, which is usually 20 microliters. Make sure that there is no anticoagulant used as a specimen for this test. After adding the drop of blood, we can start the timer and observe for the occurrence of the dimpling or the extrusion. This dimpling or extrusion is the visible nipple-like protrusion that forms on the surface of the blood drop. Reference range for this test is 15 to 45 minutes. Less than 15 minutes results may mean that there is thrombotic tendency for the patient, and more than 45 minutes is hemorrhagic tendency. An example of these incomplete or delayed results may be thrombocytopenia and Glanzmann thrombasthenia. Abnormal characteristics may be seen on the extrusion of the blood. If it's milky, the patient might have leukemia, diabetes, or may be undergoing medication. Dark yellow extrusions is jaundice, while cloudy extrusions is multiple myeloma. The qualitative Stefanini method is also known as the Stefanini-Dameshek method or the test tube method, or also sometimes as the single test tube method. This uses 3 to 5 ml of blood placed inside a Wassermann tube incubated at 37 degrees Celsius for 1 hour. After this hour, we transfer the tube at room temperature and give the reading for the first hour. After giving the reading for the first hour, we continue incubating at room temperature, and get a reading at the second hour, 16th, 18th, and 24 hours. The reference value for this test is that the retraction should begin at 1 hour and it should have been completed at 18 to 24 hours. Results for this test may be a complete retraction, partial retraction, poor, very poor, or no retraction. The McFarlane method is a quantitative test that we will be performing in the laboratory. You are to collect 5 ml of blood and dispense it into a graduated centrifuge. After dispensing, place an applicator stick in the middle, and to keep this in place, use a parafilm or a cotton. After placing the applicator stick, incubate the test tube at 37 degrees Celsius until it coagulates. And to make sure that you do not miss the coagulation of the blood, check the test tube every 5 to 10 minutes. Once coagulation is seen, transfer the test tube into room temperature and wait for retraction. Again, retraction is when the clot has retracted or has drawn itself from the edges of the test tube. Once the clot has retracted, remove the clot by pulling the applicator stick, and this clot should then be described according to its size and firmness. Size may be small, medium, and large, and normal clots would have a description of medium. Large clots would mean that there is fewer serum that is left inside the test tube. For firmness, normal clots would appear elastic, firm, dry, and they take the shape of the container, while abnormal clots are gel-like. After removing the clot, centrifuge the test tube to check for the RBC fall out and the serum drip out. A total serum drip out is known as the serum expressed, and this can be measured after centrifugation directly against the calibration of the calibrated tube. Again, serum expressed refers to the serum that was left on the tube after removing the clot. RBC fallout refers to the red blood cells that have fallen from the clot. After measuring the serum expressed, we now compute for the percent clot retraction using this formula. The amount of serum expressed, which was taken from earlier, is divided with the total volume of blood, which is 5 ml, multiplied to 100, and you will get the percent clot retraction. Normal value for this test is 40 to 60%. These are considerations that are done when performing CRT. Platelets are directly proportional to retraction. So for example, there is a decreased number of platelets like in thrombocytopenia, or when the platelets are defective like in thrombasthenia, there would also be a decrease in the retraction itself because the platelets are needed to retract the clot. In this case, the time that is needed to have a proper or a complete clot retraction would be prolonged. Thrombostanin is a protein that is released by the platelet that is responsible for clot retraction. Other contractile proteins would be actin and myosin. Other conditions that can give a prolonged clot retraction time would be Glanzmann's thromboasthenia, DIC, and PCV. PCV stands for polycythemia vera, where there is an increase in red blood cells. When there is too much red blood cells in the clot, the platelets would have a hard time shrinking the clot, leading to prolonged CRT. Therefore, we can say that the red cell mass or the hematocrit is also inversely proportional to clot retraction. Another condition is with fibrinogen. Fibrinogen amounts are also inversely proportional to CRT. Fibrinogen deficiency, like in hypofibrinogenemia or dysfibrinogenemia, can prolong CRT. And that is all for this video about clot retraction time. Thank you very much for watching.